Okay, guys, let's continue our discussion of gluconeogenesis. Now, remember the last time I pointed out this is a pathway, it's a fundamentally anabolic pathway that we have to have in order to make glucose for those tissues that will only utilize glucose as an energy source, right? That really the only thing we can supply them with is that aldohexose. So we have to be able to create glucose from non-carbohydrate sources, 3-carbon, 4-carbon backbones that aren't coming from carbohydrates. Okay, and what you're going to see is that in organisms like us, that carbon flux is primarily coming from lactate and amino acids. There are some other ways to do this, but in mammals, those are the two main sources. Okay, now there are a couple of big themes I want you to remember about gluconeogenesis as you sort of slot this into the framework of the pathways we've been talking about, glycolysis and TCA cycle. And the first big theme is sort of the centrality or the importance of a dicarboxylic acid you've already seen, and that's of course oxaloacetate, right? And we're going to see how this works, but oxaloacetate, right, is a absolutely central, whoops, look at me, absolutely central intermediate for gluconeogenesis. In fact, a lot of what's happening are the pathway is going to sort of begin in many ways, not exclusively. I'm going to back up one step, but the ability to convert other things into stuff that can be turned into oxaloacetate is sort of a key node in how gluconeogenesis works, right? Okay. To sort of emphasize that a little bit more, take a look at some of the ways in which we can turn things into oxaloacetate. Of course, we can turn pyruvate into oxaloacetate, right? Which is something we're going to see here in just a minute, right? And as I mentioned, one of the main fluxes in this pathway is going to be coming from lactate. Remember, lactate was that fermentation reaction where we took pyruvate that was coming out of glycolysis and dumped the electrons back in, dumped those NADH equivalents back into that carbonyl, turn it into an alcohol. Now we're going to, in principle, we're going to talk about how one, in principle, might want to reverse that, right? So there's a major flux from pyruvate into oxaloacetate. But we've seen some other ways one can make oxaloacetate, of course, right? So I should mention, one of the other principal ways, I said, other than lactate, where you get carbon equivalents into gluconeogenesis, of course, is through catabolic breakdown of amino acids, right? And alanine, cysteine, glycine, serine, threonine, and tryptophan, their catabolic breakdown ultimately is going to converge on the production of carbon backbones that are pyruvate, right? So they're broken down in a way that ultimately yields pyruvate, which can then be fed into the gluconeogenic pathway, right? Now, in this paper we talked about from Craig Thompson's lab, we saw another way that you can make oxaloacetate by deaminating aspartate and asparagine, right? They're 1 and 2 deamination reactions away from oxaloacetate. So breaking down, catabolic breakdown of those amino acids is another way to feed this pathway. And then, of course, catabolic breakdown, as we saw, is a big part of that paper as well. Glutamine and glutamate, through, again, a couple of deamination reactions, feeds alpha-ketoglutarate. And there are other ways. Arginine breakdown, histidine and proline also feed the alpha-ketoglutarate pool. Isoleucine, methionine, and valine are directly fed in the succinyl-CoA pool. And phenylalanine and tyrosine breakdown will directly feed the fumarate pool. So by direct access into the oxaloacetate pool from deamination of aspartate, or through pyruvate, or by taking amino acids, the catabolic breakdown products of amino acids, and feeding them into the TCA cycle, all of these things can feed the oxaloacetate pool, right? That it can be bled then for creating glucose. Okay, so oxaloacetate, big deal in thinking about gluconeogenesis. Okay, the second big deal in thinking about gluconeogenesis, that's sort of like, big theme number 1 to keep in mind. Number 2 is you've seen it before. Right? Or at least most of it, right? What you're going to see is that gluconeogenesis, by and large, as we talked about last time, is this reversal right? And we know that it can't be a perfect reversal, but the pathway design for gluconeogenesis is in that second category, right? Because if you remember, a large number of the steps operating down this pathway are operating very close to equilibrium, right? In other words, they have free energy, standard state free energies that are positive, and cellular conditions just manages to pull or push them in the sort of negative delta G prime, push that cellular condition free energy in just the negative direction. So those steps are just hanging out barely away from equilibrium, which means they can be easily reversed. So all we need is a couple of key steps that we can control. And if you remember where those key, largely exothermic steps are in glycolysis, they're up here at the beginning and down here at the bottom, right? At the hexokinase or glucokinase reaction, right up there at the beginning, the phosphofructokinase reaction where we make fructose 1,6-bisphosphate and the sugar is then committed, right? Big exothermic steps, like lots of free energy, right? And then down here at the pyruvate kinase reaction, we take phosphoenolpyruvate and we actually make pyruvate. All of those intervening steps between phosphoenolpyruvate and fructose 1,6-bisphosphate, if you remember, are operating just barely negative in free energy or just a bit negative in free energy under cellular conditions, or positive in free energy or close to zero, so they have to be pulled or pushed in the glycolytic direction, which means these steps are all easily reversible. But that also means the chemistry of all these steps is simply the exact opposite of what you saw when we talked about glycolysis, right? So for this to work then, in the context of our last discussion, all we need are bypass reactions for the beginning of this pathway and the end of the pathway that we can coordinately control, right? So we need some alternate steps over here that bypass, can do this same reaction in terms of taking pyruvate and making phosphoenolpyruvate in a different way, and then ways in which we can take 1,6-fructose 1,6-bisphosphate and then turn it back into glucose with a different set of enzymes, right? So we have to be able to bypass, as we talked about, and then coordinately control. So today, the rest of the lecture, we're going to talk about the chemistry of these steps, how we build these bypass reactions, what the chemistry and the enzymes do. And then in the next lecture, we're going to talk about how this regulation is coordinated between running this set of reactions or this set of reactions and making the pathway go one way or the other. Okay, so those are the two big themes, right? So let's dive right on in here and take a look at how this fascinating sort of in first step of this pathway works. All right, so what I'm going to call this reaction 1 of gluconeogenesis, and this is a bit of a misnomer because the enzyme that we're going to use here to turn pyruvate back into oxaloacetate is doing this not just to run glycolysis. It's basically doing this to maintain the oxaloacetate pool. But that, as I pointed out, that's the pool you tap when you want to make glucose, right? All right. So remember, we're going to start here now with pyruvate, right? We're going to take some ATP. We're going to take some carbon dioxide and some water. And we're going to basically run a carboxylation reaction. We're going to take that CO2 group and we're going to stick it, take this CO2 group here, and we're going to stick it onto that methyl group right there, all right? So to create, of course, oxaloacetate, right? Cool, that's neat. All right, so I should also mention, of course, this is going to burn some ADP, right? We're going to create some, you're going to burn an ATP to create ADP and inorganic phosphate in the course of doing this, right? So oxaloacetate, and here we've got pyruvate, right? The enzyme that runs this reaction is, of course, pyruvate carboxylase. And important, there's some really interesting wrinkles to this reaction. One is that this enzyme requires a cofactor you haven't met yet, right? It's called biotin, right? And biotin is going to be absolutely essential for activating that CO2 group so that we can synthesize that carbon-carbon bond in oxaloacetate. Okay, there's another interesting wrinkle. You remember the last time we talked about this, that when you think about glycolysis and gluconeogenesis and this idea of feudal cycles, the fact that the glycolytic and gluconeogenic enzymes are all in the same place, they're all in the cytoplasm, we have to have that coordinate control, right? Okay, there's one exception to a critical enzyme for gluconeogenesis that's not in the cytoplasm. That's this guy. Pyruvate carboxylase is actually in the mitochondrial matrix, right? So it's in exactly the place where TCA cycle is running, otherwise using or making oxaloacetate. And it's really important there, right? The pyruvate carboxylase is an important enzyme, both in terms of keeping the TCA cycle running, right? By being able to take pyruvate equivalents and turn them into oxaloacetate. But by feeding that oxaloacetate pool, it's also a critical enzyme for gluconeogenesis, right? So it makes some sense why pyruvate carboxylase would be in that place. But that creates a problem that we'll talk about here in a second. Now, something just occurred to me. Yes, I made a mistake here. Let me correct it as I was writing this down. I don't know why I had that as an alcohol. I started to write it correctly. Let me just go ahead and fix it. Oxaloacetate. Whew. Okay. All right. So we've taken, we're going to take pyruvate, and somehow we're going to create this carbon-carbon bond. Well, you can kind of almost begin, if you remember some of the things we've talked about before, about how this might work. Remember, right, what I've told you guys about before, reminded you of, about the acidity of those protons that are methyl to that carbonyl group, right? Their acidity and the fact that, you know, if you had a good base, right, you could abstract those guys, drive those here, and you've got the resonance available in that carbonyl oxygen to create that enol, right? And that enol is going to be key to how this reaction is going to go. But what about the ATP? What do you need the ATP for? Right? And the biotin, what's that for? All right, so let's take a look at the mechanism of this reaction and see how this runs. All right, so the pyruvate carboxylase reaction, you can sort of think of as happening in two steps, really, when you get down to it, or two phases, right? There are two parts to it. It's all in the same enzyme, but it's sort of in two halves. The first half is we need to activate some CO2, and that's what the ATP is for. You guys remember, hopefully, that when you have CO2 in aqueous solution, the vast majority of it is tied up in bicarbonate, right? And that bicarbonate's not very reactive. That's not reactive at all, right? That carbon's not very electrophilic in this sort of hydrated state, right? So what the ATP is for is to essentially dehydrate the CO2 group. And you do that by, in the active site, or one active, this enzyme actually has two active sites. In one active site of the enzyme, you get that bicarbonate to react with this, one of the phosphate groups of the ATP to create so-called carboxyphosphate group, right? Now, that is essentially, that reaction is driven by the favorable free energy change of hydrolyzing this ATP, all right? But that carboxyphosphate's not stable, right? It's a very unstable molecule. And if you were just doing this out in free solution, the CO2 would come off, right? Just like it's sort of electron pushing shown here. You'd split that carboxyphosphate group and it would just rehydrate again, right? But in the active site of the enzyme, where it's now changed conformation, as we've talked about before, and it's dehydrated the surface, dehydrated the interior of the active site, there's something else there that's ready and waiting for the CO2 group, and that's this biotin group. So biotin is this interesting molecule. If you imagine taking sort of urea, a couple of nitrogen groups, and with a carbonyl in between them, and then you put on the other side of that this sort of five-membered sort of thioether-like compound, you get the double ring structure of biotin, right? And it's connected by this group little four methylenes through an amide bond to a lysine residue. And so it's covalently attached in the active side of the residue, active side of the enzyme. And it's actually on a fairly decent little arm. This is sort of extends out. It's like a little 14 angstrom swing arm. I've talked about swing arms before. Remember succinyl-CoA synthase? right? Or pyruvate dehydrogenase complex or alpha-ketoglutarate dehydrogenase complex. This swing arm thing is like a little theme, right? Well, this biotin's out on the end of a little swing arm. And when the carboxyphosphate is formed in one active site of the enzyme, this sort of extended swing arm reaches over into the active site where the carboxyphosphate is formed. And as the carboxyphosphate decays, one of those nitrogen groups can attack that carbon of that CO2 group to form so-called carboxybiotin, right? So here's that swing arm, all right, on the end of the domain where it's attached to that lysine residue. And you get this activated CO2 group in this carboxybiotin, right? So that keeps it stably activated, electrophilic, and dehydrated long enough that the biotin swing arm can now move over to the other part of the enzyme, the other active site, where the actual condensation reaction happens, right? So here again is that carboxybiotin. And in the other part of the enzyme active site, we've now, or the other active site, we've now bound pyruvate, right? Now what we need is a good electrophile. And remember, I mentioned a minute ago, the acidity of those protons that are alpha to that carbonyl group. So we know with a little acid-base chemistry, we can pop that baby off, right? And create an enol, right? And I think this is somewhat controversial. There's some evidence that the biotin itself might act as the base to help drive that reaction, and you actually generate temporarily gaseous CO2 in the active site of the enzyme. I think there are other biotin carboxylating enzymes where you actually have the enol attack the carboxybiotin itself, right? So whichever mechanism is exactly right in the case of pyruvate carboxylase. I don't know if this has been settled or not. People argued about this. In any event, this activated CO2 group then gets acted upon or nucleophilically attacked by that enol to then synthesize oxaloacetate, right? So we've created that carbon-carbon bond, which is great. That's awesome. We made oxaloacetate. And as I said, this is a super important thing going on in the matrix of the mitochondria, right? Because if your oxaloacetate pools collapse, you can't run TCA cycle, right? So pyruvate carboxylase is hanging out in here in a really important way, taking pyruvate, as you recall, being made by glycolysis, breakdown of our carbohydrates. It's imported into the mitochondria, ultimately can be used, through pyruvate dehydrogenase in the TCA cycle, but it can also be bled, and it's important, some fraction of it is bled and fed by pyruvate carboxylase into the oxaloacetate pool to make sure those pools stay where they need to be. But if you don't want to run your carbon equivalents, you've run out of glucose, right? You don't have glucose to run. And what you've got is amino acids. And let's say your liver tissue or kidney tissue, and one of your big jobs is to regulate blood glucose levels. You need to take some other carbon equivalents and turn them into glucose, which means you need to pump this oxaloacetate pool using pyruvate carboxylate and then turn it into glucose. Okay, but here's the problem. Like I said, this is all happening in the mitochondria. Guess what does not exist in the mitochondria, right? The gluconeogenic enzymes are all out here in the matrix. There is no transporter for oxaloacetate in the mitochondria. There is no way to directly import or export oxaloacetate. So all this oxaloacetate that's being made under conditions where you're going to downregulate TCA, you're going to glycolysis is shutting down. You need to pump this flux back into making glucose. You've made something you can't get out of the mitochondria directly. Okay, so this is the wrinkle in how this works. You have to convert it into something that you do have a transporter for. And there are two main ways this happens. One involves taking that oxaloacetate, running the malate dehydrogenase reaction the way we would run it through TCA, as we've talked about, right? Running the malate dehydrogenase reaction sort of in reverse, right? Because remember, we would normally go from malate to oxaloacetate, but if you reverse it, you take some NADH, you reverse that enzyme, you take the oxaloacetate, you convert that carbonyl group back into an alcohol to make malate. There is a malate shunt, right? So you can now take that malate and you can shuttle it out into the cytosol. And guess what happens to exist in the cytosol? There's also a cytosolic malate dehydrogenase enzyme, which will do the exact opposite. It'll do what we would what we talked about before or looked at before, running glycolysis, running TCA cycle, it'll take NAD plus and convert the malate back into oxaloacetate, convert the alcohol group back into a carbonyl. Now this does two things. It both moves oxaloacetate from the matrix, the mitochondria, into the cytosol. It also moves a reducing equivalent, right? Because you pump some electrons in here into malate by using an ADH, and here you suck them back out. So this is one of the ways to move reducing equivalents out of the mitochondria into the cytoplasm at the same time that you're moving oxaloacetate equivalents out. There's another way to do it, which is related to the discussion we had today, that paper from Craig Thompson's lab. You can also simultaneously move activated nitrogen groups, right? So if you take oxaloacetate and instead of reducing it to an alcohol, you amidate it, right? You use a transaminase reaction and you stick an amino group on it. There's also an aspartate shunt that will move aspartate equivalents in and out of the mitochondria. And there is an aminotransferase in the mitochondria that will then take aspartate and convert it back into oxaloacetate, right? Simultaneously moving out an activated nitrogen for you as well. Okay, that's how we get started. This is this important sort of centrality of oxaloacetate. In the next video, we're going to see how we then take oxaloacetate in the cytoplasm and turn it into glucose.